Welcome to the lecture cast on specialized oral mucosa. Oral mucosa is the moist mucous membrane that lines the oral cavity. It performs the function of protection, sensation, secretion, and thermal regulation. Oral cavity consists of two parts, the vestibule and the oral cavity proper. Vestibule is confined by the lips and the cheeks, whereas the oral cavity proper has the superior zone as the hard and soft palate. Inferiorly, it is bordered by the floor of the mouth and base of the tongue. Posteriorly, it has facial pillars and the tonsils. Oral mucosa is distinct in different parts of the oral cavity. Based on the primary function, it can be categorized into masticatory mucosa, lining mucosa, and specialized mucosa. The lining mucosa mounts to 60% of the oral mucosa. It is distendable and relatively loosely bound to the adjacent structure. Floor of the mouth, cheek, lip, and soft palate are the examples of lining mucosa. Masticatory mucosa constitute about 25% of oral mucosa. It is rigid, tough, and tightly bound to the underlying bone by means of dense connective tissue. Hard palate and the alveolar ridges are the example of masticatory mucosa. Specialized mucosa comprise of 15% of the oral mucosa. It has specialized mucosal structures like lingual papilla and the taste receptors. Dorsum of the tongue is the characteristic example of specialized mucosa. Histologically, when you take the tissue sections of the oral mucosa, you find two distinct tissue components. They are the overlying epithelium and the underlying connective tissue. The overlying epithelium is classically the stratified squamous epithelium, which is stacked into different layers. Based on cornification, they can be a keratinized, which is seen in the masticatory mucosa, and a non-keratinized, which is seen in the lining mucosa. The underlying connective tissue is divided into lamina propria, which bears the grunt of the overlying epithelium, and the submucosa, which connects the epithelium with the deeper structures. Histology of specialized oral mucosa is also similar. It has got the overlying epithelium and the connective tissue core. There are taste buds which are interspersed into the keratinized layer of the epithelium, which opens to the exterior with means of taste pores. The mucosa of the dorsum of the tongue, it is unlikely anywhere else in the oral cavity. Why is it so? Why is it so specialized? Although it is being lined by functionally a masticatory mucosa, it has also got a highly extensible lining. In addition, there are presence of lingual papilla. These lingual papilla has the masticatory function. They are also provided with some taste buds, thus performing a sensory function too. The mucous membrane of the tongue is divided into two parts by means of a V-shaped sulcus called sulcus terminalis or terminal groove. The anterior two-thirds of the tongue, which is derived from the first pharyngeal arch, forms the body of the tongue, and the posterior one-third derived from the third pharyngeal arch forms the base of the tongue. Lingual papilla and the taste buds are commonly present in the anterior two-thirds of the tongue. There are various lingual papilla present on the tongue, which are the fungiform, filiform, circumvallate, and foliate papillae. Regarding the distribution of these papilla, the anterior two-thirds has the filiform papilla, which is completely present in them. The fungiform papilla is almost present at the tip of the tongue. The foliate papilla is present on the sides of the tongue, whereas the circumvallate papilla are arranged adjacent or anterior to the sulcus terminalis. At about 7th week of fetal life, the valate and the foliate papilla develop very close to the terminal branches of the glossopharyngeal and the vagal nerves. By 10th week of fetal life, the fungiform and the filiform papilla cover almost entire anterior two-thirds of the tongue. They are seen developing near the terminal branches of chorda tympani, which is a branch of facial nerve. Coming into details of each individual papilla, we can see a fungiform papilla which is mushroom shaped. Due to the rich vascular network, large size, and the sparse distribution, it has been identified as reddish dots. Each individual is estimated to have about 200 fungiform papilla. They are so numerous or denser at the tip of the tongue than the middle region. They are scattered in between the numerous filiform papillae. They have a smooth, round structure. The reddish appearance is due to the high vascular connective tissue core. The epithelium is thin, non-keratinized stratified squamous epithelium. Taste buds are present on the superior surface. On an average, they have 2 to 4 taste buds. Some of the fungiform papilla don't even have taste buds. The fungiform papilla count is used to estimate the taste sensitivity. The picture shows the histological appearance of fungiform papilla. You have a overlying epithelium and your underlying connective tissue core, and the electron microscopic picture shows the fungiform papilla surrounded by numerous filiform papillae, and you have a taste pore which is opening into the exterior amidst the surrounding epithelial cells. The filiform papilla is a hair-like conical structure which projects 2 to 3 mm high from the surface of the tongue. It covers almost the entire anterior part of the tongue. It has got a tough abrasive surface. When food particles are being compressed against the palate, it facilitates the breakdown and the masticatory process. The epithelium is thick keratinized stratified squamous epithelium. The keratinization is to withstand the masticatory stress. Some of the individuals, the buildup of keratin and the failure of desquamation of these keratin leads to the elongation of the filiform papilla, giving a hairy appearance to the tongue. The taste buds are not present in the filiform papilla. They provide for the touch, temperature, and the pain sensations. The picture shows the histological appearance of the filiform papilla. You can see the hair-like projections of the papillae. The keratinized projections are interspersed with the non-keratinized areas. While the keratinization helps in to withstand the masticatory forces, the non-keratinized part, it helps in the extension of the tongue. Moving on to the circumvallate papilla, these are large papilla, 10 to 14 number. They are arranged in a row and adjacent and anterior to the sulcus terminalis. They are at level with the surface of the tongue. These have a characteristic walled appearance. They are circumvallate. They are surrounded by wall on all sides. The walled appearance is due to the deep groove which surrounds the papilla. The duct of the minor salivary gland, also known as the von Ebner's gland, open into the groove. The secretions, which are very serous, helps to flush the food particles deposited in the groove. The superior surface of the papilla is keratinized stratified squamous epithelium, whereas the lateral portions are non-keratinized. These lateral walls also has the presence of numerous taste buds. The picture shows the groove surrounding the papilla, and the histological picture shows the keratinized superior surface and a non-keratinized part on the lateral surface. You can see the taste buds studded on the lateral wall. It extends to the entire thickness of the epithelium. Foliate papilla, these are leaf-like papilla which are present in the lateral margins of the posterior part of the tongue. They are frequent in mammals other than humans. These are pink-colored papilla. 4 to 11 parallel ridges are alternating with deep grooves. These are non-keratinized stratified squamous epithelium. which is covered the entire papilla, and this has got also a connective tissue core. There are numerous taste buds present on the lateral walls of the ridges. This, like a circumvallate papilla, the serous glands also surround under the taste buds, which serves to cleanse the trencher of the papilla. The picture shows the ridged appearance of the foliate papilla. The histologically, it has got a non-keratinized epithelium and with the lateral walls showing the taste buds. What are taste buds? These are the organ of taste sensation. They perceive the taste sensation. They are seen developing at about 11 to 13 weeks of fetal life. The evidences show that the taste responses develop as early as 26 to 28 weeks of gestation. On an average, an adult shows 30,000 to 10,000 taste buds. Children have more than that. Beyond 45 years, the taste buds degenerate, leading to a decreased taste sensation in older age. The taste buds are more predominantly found associated with the lingual papilla of the tongue. They are also present in soft palate and pharyngeal area. The histological picture showing the distribution of taste buds in different papilla. They are seen in the superior surface of the fungiform papilla, whereas they are also present in the lateral aspects of foliate and the circumvallate papilla. The taste buds are the barrel-shaped bodies composed of 30 to 80 spindle-shaped cells. They are closely associated with sensory nerves, thus they are called as the neuroepithelial structure. If you see a taste bud, it has got a basal portion which is being delineated from the underlying structure by means of a basal lamina. Apically, they end into a taste pit. The taste pit opens to the exterior as a taste pore. Thus, there are cells, taste cells. These taste cells can be a supporting cell which are found in the periphery of the taste bud, and some of them are the sensory cell, which are the cell of taste transduction. These sensory cells, they extend their small projection, which are also called as the microvilli, into the taste pore. There are some cells which are called as the basal cells. These do not extend to the entire thickness of the taste, but these differentiate into the new receptor cells. These are said to be derived from the surrounding epithelial cells. The apical ends are tightly bound by means of junctional complexes. Based on electron density, the taste cells are classified into type 1, type 2, type 3 and type 4. Type 1 cells are highly dark. These are abundantly present cells. They extend their cytoplasmic extensions or the lamellae that engulf the other cells. They exhibit ionic currents that are implicated in the salt taste transduction. They express GLAST, a transporter for glutamate. They are involved in terminating the synaptic transmission, restricting the spread of transmitters. Thus, they act as glial cells of nerve or brain. The type 2 cells, they are morphologically similar to type 1 cells. They have in them the embedded receptors for sweet, bitter and umami compounds. These receptors are G protein coupled receptors with 7 transmembrane proteins. They are the taste sensing cells. They express voltage gated sodium and potassium channels essential for producing action potential. They also have hemichannel subunits, which are the key players in taste evoked secretion of ATP. The type 3 cells are those cells which express proteins associated with synapses. Thus, they are called as presynaptic cells. They form synaptic function with the nerve. Apart from the neuronal function, the type 3 cells also have receptors for sour taste and carbonated solutions and are presumably the cell responsible for signaling these sensations. The type 4 cells are undifferentiated cells which are present near the basal lamina, which contain the intermediate filament, and they are said to be derived from the surrounding epithelial cells. These cells, when need occurs, differentiate into the taste cell receptor cells. There are 5 basic taste perceptions. They can be sour, bitter, salt, sweet, and umami taste. These 5 types of taste sensation are detected with evidence of regional sensitivity. That is, the sweet and the salt taste are perceived at the tip of the tongue, the sour taste on the side of the tongue, and bitter taste on the region of valid papillae. The areas of particular taste sensation overlap. All papilla are responsive to all types of taste sensation. The level of sensitivity differs. Mixing of 5 modality taste along with other taste sensations like that of fatty and metallic taste sensations, coupled with odor and temperature, produces all the flavors we experience. The taste receptors, these are the chemical receptors which are studied into the taste cells. There are about 13 chemical receptors in taste cells which are identified. Sweet, bitter, and umami taste cause transduction mediated through G protein coupled receptors. The sweet taste are hardwired to T1R2 or T1R3 receptors, umami taste T1R1 and T1R3 receptors, and a bitter taste are transducted to T2R receptors. These sweet, bitter, and the umami taste receptors are found in the type 2 taste cells. The sour and the salt taste modulate function of receptor by direct activation of specialized membrane channels. They are not associated with the G protein coupled receptors. The PKD channels, which are found in the foliate and the valid papillae, mediate the sour taste. Along with them, the acid sensing ion channels which are present in the fungiform papillae also help in the mediation of sour taste. The salt taste is mediated by epithelial surface sodium channels. The salt taste receptors are present in the type 1 cells, whereas the sour taste receptors are found in the type 3 cells. Moving on to the taste pathway, when a food is being taken inside and when they get broken down, the molecules go and occupy the taste pit, the microvilli. They move the molecule towards the taste receptors and they get absorbed onto the membrane receptors. As soon as they get conformed to the taste receptors, there is a conformational change which changes the ion flow across the taste membrane. The activation of the signaling cascade through the downstream molecules happen with the help of membrane associated proteins like transducin and gusteusin, ultimately causing a change in the membrane depolarization and the release of transmitted substance, mainly serotonin, thus activating the unmyelinated afferent nerve fibers. Associated nerves are the facial nerve, glossopharyngeal nerve, and vagal cranial nerve. These rely in the higher centers for taste perception. The higher centers are solitary nucleus present in the medulla oblongata, ventral posterior medial nucleus found in thalamus. Finally, the gustatory cortex. You have a place called insula in gustatory cortex which consciously perceive the taste sensation and finally stores the taste memory. These are the references for further study. Thank you.